Welcome to the AQuA project. AQuA is a citizen science project bringing together communities to better understand the health of our rivers and lakes. Working together, volunteers collect water samples and environmental observations using the same methods and equipment at locations across different freshwater environments. This shared approach is one of the things that makes AQuA unique. By community groups collecting samples in the same way, we can combine results from many different rivers and lakes and build a much larger and more powerful picture of water quality than would be possible at any one site alone. The methods used in the AQuA project have been developed and tested with our project partners, including the Environment Agency, to ensure that the data collected are reliable, accurate, and can support our understanding of freshwater environments. The samples collected help us measure a range of water quality indicators, including nutrients, bacteria, metals, and organic chemicals such as pharmaceuticals. In this video, we'll walk through the full process of collecting water samples for the AQuA project, from preparing to go out into the field, through to collecting samples safely and bringing them home for analysis. You do not need previous scientific experience to take part. The toolkit is designed so that community groups and volunteers can collect high-quality environmental data and help us better understand water quality in your local rivers and lakes. Before going out sampling, there are a few important things to remember. First, always read the project risk assessment carefully before sampling. You'll also be asked to confirm that you've read and understood it in the AQuA Data Collection app. Never undertake sampling alone. Wherever possible, work with at least one other person and carry a fully charged mobile phone. Only collect samples during daylight hours and check the weather forecast before leaving home. Do not sample during thunderstorms, flooding, high winds, or other hazardous conditions. Wear appropriate clothing for the weather and terrain, including sturdy footwear with good grip. Depending on conditions, you may also need sun protection, drinking water, or warm layers. Never enter the river or lake. All samples can be collected safely using the bucket and string provided. If you can't safely reach the water from the bank, a bridge, or another safe access point, do not attempt to collect a sample. When sampling from bridges, remain behind barriers and never lean your upper body over the edge. When sampling from riverbanks, ensure the ground is stable and be particularly careful around mud, slippery surfaces, steep banks, and uneven ground. Be aware of your surroundings at all times, including traffic, cyclists, wildlife, members of the public, overhead power lines, and vegetation. Surface water may contain harmful microorganisms and other contaminants. Avoid touching your face while sampling and wash your hands thoroughly after completing your fieldwork. Consider bringing hand sanitizer along. Your safety is always more important than collecting a sample. If at any point you do not feel safe, postpone your sampling and return another day. Your AQuA kit contains two separate boxes. The first box contains everything needed for field sample collection. The second box contains materials for sample processing and analysis back at home or in another clean and covered area. Inside the field sampling box, you'll find several small paper bags. Each paper bag contains everything you need to collect samples from one sampling site. Inside, you'll find materials to collect 4 different types of water sample. First, a small plastic vial for measuring E. coli. Second, 2 amber glass vials, a syringe and a small filter for measuring organic contaminants such as pharmaceuticals. Third, a plastic bottle, larger syringe and larger filter for measuring metals. And finally, 2 plastic tubes, a larger syringe and large filter for measuring phosphate and other nutrients. Your kit also contains labels for each site and a list of locations to visit. Before starting at a site, check that you're using the correct labels for that location. If you'd like, you can apply these labels to the vials at home before heading out to collect the samples. You will also need to keep the samples cool in the field after you collect them. This prevents them from degrading before analysis. To do that, you'll find some small dehydrated ice packs in your sampling kit. The day before you set out, be sure to soak the packs in water to hydrate them and freeze them overnight. When you arrive at your sampling location, begin by finding a safe and stable place to work, from the riverbank or on a bridge with a pedestrian walkway. Avoid steep, slippery or unstable ground. Before collecting samples, prepare your equipment and put on gloves. Keep your sample bags and labels organized throughout the process. Your samples should be kept cool while in the field using the ice packs provided. To collect some water, simply lower the bucket beneath the water surface. Avoid disturbing sediment at the bottom of the river where possible. The first bucket of water is used to rinse the sampling equipment itself. Discard this water safely onto the bankside vegetation or back into the river or lake. Then collect a second bucket of water full to the top of the bucket. This bucket will be used for sample collection. If you run out of water while collecting your samples, just lower it back down to collect some more. For the pharmaceutical and personal care product sample, you'll fill 2 glass vials with 2 milliliters of filtered water in each. To do this, first draw the water into the 3 milliliter syringe, then empty it to rinse before taking your sample. Then refill the syringe and attach the small 13 milliliter glass microfiber filter to the syringe by screwing it on. This filter will remove bacteria that may break down the chemicals in the water and small particulate material which may interfere with sample analysis. First, push the syringe plunger down to fill the glass vial with about 1 milliliter of water. Rinse the vial with this water and discard it. Then slowly fill the vial with the remaining 2 milliliters of water in the syringe. Do not put more than this in the vials, as the water will expand when frozen and burst the vial, making the sample no longer usable. Secure the cap tightly and attach the correct site label. You can also attach the label before going out in the field if you like. If the filter becomes difficult to push through, replace it with a spare filter. Repeat this process for the other small amber glass vial in your sampling kit. Once you've finished and ensured the samples are labelled, place them back in the paper bag for that site and put this on the ice packs to keep cool. This prevents the samples degrading while you're in the field. After you collect this sample, it will be sent to the University of York lab, where it will be analysed for organic chemicals like pharmaceuticals and personal care products that may be in the water. Sometimes these types of chemicals can have impacts on aquatic organisms, so it's very important we know how much is in the water. For the nutrient samples, you'll fill 2 plastic tubes, one for the HANA phosphate measurement you'll do at home, and one for analysis of nitrate, nitrite and ammonia in the university lab. To do this, use the larger 10 milliliter syringe and larger 25 millimeters nylon filter. Draw water into the larger 10 milliliter syringe and attach the filter by screwing it on. Rinse the sample tube first using a small amount of filtered water and discard it. Then filter 10 milliliters of river water into the sample tube and secure the cap. You'll then repeat this process for the second plastic tube. Be sure the correct labels are applied to the tubes, then keep the samples cool with the ice packs. Nutrients can enter rivers and lakes from sources such as agriculture, wastewater, urban runoff and decaying organic material. At high concentrations, nutrients can contribute to poor water quality and affect aquatic ecosystems, so monitoring them helps us better understand river and lake health. Before you collect the sample for metals, be sure you've read the risk assessment document. The bottle used for this sample contains a very small amount of acid, no more than a drop, which helps preserve the sample before laboratory analysis. The acid is low risk when handled carefully, but it may cause skin irritation, temporary discoloration or discomfort if it comes into contact with skin or eyes. For this reason, please always wear gloves and eye protection when handling the open bottle, and always angle it away from yourself and others. To collect the metal sample, fill the large syringe with water from the bucket, then attach a large syringe filter by screwing it on. You do not rinse this bottle before use. Keeping the bottle angled away from yourself and others, slowly push the syringe plunger to filter 10 milliliters of water into the bottle. Once finished, securely fasten the lid and check that the correct label has been applied. After collecting the samples for metals, it will be sent to the University of York for analysis. Metals can enter surface waters from sources such as road runoff, industry, mining and urban drainage. Some can affect aquatic organisms at high concentrations, so it's important to study them in surface water. To collect the E. coli sample, fill the sterile plastic vial directly from the sample bucket. Do not use filtered water for this sample, as the filters remove bacteria from the water. Secure the lid immediately and apply the correct site label. After collecting the E. coli sample, you will bring it home and use the AQuA E. coli test to grow and measure any E. coli bacteria that may be present in the water. E. coli is commonly used as an indicator of fecal contamination and can help us better understand potential sources of microbial pollution in rivers and lakes. In some sampling kits you'll find materials to collect a field blank, or also called a quality control sample. This won't be required every time you sample, but if your kit does include field blank sample materials, please complete this step as part of your sampling. Quality control using a field blank sample helps us check that the methods are working correctly and ensures we produce the most accurate and useful results possible. For this sample, you'll be provided with a bottle of clean water. This is specially prepared water that does not contain the bacteria, nutrients, metals or organic chemicals we're measuring in the environment. First, pour some of the clean water into your sampling bucket. Swirl it around to rinse the inside and then discard this water. This cleans the bucket and helps prevent contamination from any previous samples. Next, refill the bucket using more of the clean water. Now collect your field bank samples following exactly the same steps used for your river or lake samples. Your kit will tell you which quality control samples to collect. You may only be asked to collect field bank samples for some sample types such as E. coli and pharmaceutical or personal care products rather than all of them. By testing water that we know does not contain these contaminants, we can check whether anything has been introduced during the sampling process itself. These important quality checks help make sure the results collected by AQuA volunteers are reliable and can be used with confidence. At each site, be sure to record your observations in the AQuA Data Collection app. This includes the sampling location, photographs, environmental observations and any notes about conditions at the site. Try to complete the data entry while still at the location where possible. If you'd like detailed guidance on using the app, be sure to check out our dedicated app user guide and video. Before leaving the site, check that all bottles are labelled correctly, all samples are sealed tightly, observations have been recorded in the app, and all samples are stored safely in the paper bag for that site and placed with the ice packs. Use the sampling checklist to make sure you've completed everything. When you've finished collecting the samples, place one of the small ice packs in the paper sample bag, along with the samples themselves. Everything else can go in the waste bag provided with your kit. Keeping the samples cool prevents them degrading before analysis. You're now ready to go to the next site. Once you've finished sampling all of your sites, return home or to another indoor space. Here you will process the E. coli and phosphate samples and prepare the remaining samples for storage and return shipment. Start by taking out the paper bags containing the samples collected from each site. Remove the labelled vials for E. coli and phosphate testing and keep these separate. If you're not ready to test them straight away, place the vials in the refrigerator until later the same day. All the other samples will be shipped to the University of York for analysis. To prevent them degrading before analysis, be sure to place these samples in the freezer along with the ice packs as soon as possible. Please keep them in the freezer until you're ready to send the samples to the university. Detailed instructions for analysing your E. coli and phosphate sample, then shipping the rest to the university for analysis, can be found in the next training video. Thank you for taking part in the AQuA project. By collecting these samples, you are helping to build a better understanding of water quality and pollution in rivers and lakes. Your contribution supports both scientific research and community-led environmental action. If you need support at any stage, please contact the AQuA team or scan the QR codes provided throughout the toolkit for additional guidance videos and instructions. Thanks for watching and happy sampling.